in today's class, we will learn about the basics of the immune system. In fact, nowadays, you all may be hearing a lot that those who have a healthy immune system, they are protective from this COVID situation. However, those who have a weak immune system or a compromised immune system, they are getting the COVID. And this might have triggered the curiosity in all of you that what is immune systems? Because most of you know that or might have studied that respiratory system, gastrointestinal system, nervous system. But what is the immune system? So unlike all these systems what you heard before, especially the respiratory system, gastro, or the endocrine system, immune system is not just simply a set of organs, but instead it's a very dynamic kind of system which comprises of many different cell types, and you cannot define the immune system by one organ. So multiple organs together, multiple cellular system together, they together make the immune system. So immune system, basically in a very simple word, the system that protect our body against all or very different variety of the pathogen. Okay. It's a very important question that we all are surrounded by so many pathogens, so many microorganisms which can cause disease. We are continuously getting exposed to majority of these pathogens. For example, Mycobacterium tuberculosis, this TB bacteria, is all around us, especially in our country, for example, or other developing country. In developed world, they have a lot of fungal infections. Fungus is growing in the more like moist and cold environment. But majority of the human beings, they live normal life. So what defend our body? And that is the immune system, which is continuously fighting 24/7 against all these microbes that attack us and keep us all safe. In very simple words, immunity is basically your body's ability to resist or eliminate potentially harmful foreign materials or abnormal cells. So if you see these, what are these harmful foreign material or abnormal cells? For example, viruses, including SARS-CoV, this novel coronavirus. Toxins, which I told you in the bacteria class, that let us say Staphylococcus aureus, that releases toxins, anthrax toxin, fungus spores, for example, they can cause infection, various type of bacteria, parasites, helminth, for example, in the intestine, germs, different kind of germs, pollutants, carcinogen means the material that causes cancer. So all these different type of pathogen or the material or the abnormal cells which are causing some harm to our body, immune system continuously fight against them. Okay, so basically immune system is defense against the invading pathogens like viruses and bacteria. They remove the worn out cells. So for example, in our blood, the red blood cells which you learned, they have the hemoglobin that carries the oxygen. These RBCs get old. So worn out cells, immune system remove, and we get the generation of the more healthy red blood cells. Similarly, tissue debris. So after injury, our tissues get damaged. So all the damaged tissue should be removed from our body because new tissue will be regenerated. And that work is done by the immune system. Identification and destruction of abnormal or mutant cells. So for example, when you go on UV light or you are exposed to X-rays, many times you can't avoid exposure to UV light or exposure to X-rays or exposure to different material that can ultimately cause cancer. So our cells are continuously getting mutated also after exposure of these different material and pathogen. But still, majority of do not develop cancer. Why? Because our immune system continuously destroy these abnormal or mutant cells and keep us healthy. Rejection of foreign cells. So if, for example, you might have heard that you cannot donate simply your blood or simply your organs to the other person, because for other person, your body is foreign. So especially, for example, now you can imagine about the transplantation, organ transplant, liver, kidney, those transplanted, anybody cannot donate to anyone. You have to ensure that certain factors are matched in recipient as well as in donor. Then only organ transplant is successful. So foreign cells are always rejected by our immune system. Only when there is certain match, the foreign cells are accepted. Okay, but there's always a but in the life, right? Because majority of the time, immune system protect us from the outside and the foreign material. However, sometimes immune system can also become hyperactivated and can make mistake. And that hyperactivation can happen because of unharmful antigens or unharmful material. For example, pollens. The pollen, many of you have noticed that whenever there's a pollen season, we all get allergies. So allergy is hyperactivation of immune response against unharmful antigen or unharmful material. But sometimes our immune system gets so activated, it fights against us only. It has got somehow inappropriate immune responses. That means our body is killing our own body, and that causes autoimmune disorders. So in general, so in brief, our immune system keep us safe from the potential harmful antigen, but sometimes the inappropriate activation also occur that can cause allergies, which is against basically activation of immune response against the unharmful antigens or harmless substances. But sometimes it can get overactivated and start killing our own tissues. That is called autoimmune disorders. If you go back to the history and find out what imparts the immunity, you will find very good work by Emil von Behring and Kitasato in 1890. Actually, they found out that blood from the vaccinated animals, that blood has certain components which are protective in nature. In a slight more detail, that you know that blood has one liquid component, another is cellular component. The liquid component of the or non-cellular fraction of the blood is called serum, and that serum has the antibodies. That time they did not know that antibodies are present in the serum, but what these people found out, that if they take out this non-cellular fraction of the blood, they found this non-cellular fraction of the blood, so-called serum, is protective against the diphtheria or tetanus. So they took the animal which were vaccinated against diphtheria or the tetanus, and then they took the serum antibodies and they treated the diphtheria patients or the tetanus individual and found out that those got protective. This is very similar to today's time in the corona vaccine, corona COVID pandemic time, because some of you might be hearing this plasma therapy for the COVID-19. So what is plasma therapy for COVID-19? Plasma therapy of the COVID-19 is that if you take the plasma from the COVID patient, you are expecting that that plasma will contain the antibodies. If you use these antibodies now, you can use these antibodies to treat the another COVID patients. So ideal concept, now you can imagine that concept was there in 1890, where the antibody discovery happened, like the concept came. But similar concept is even used nowadays and very relevant for you all to appreciate that plasma therapy for COVID patients is done according to this concept. Okay. So now comes to Alan Metchnikoff. So while Emil von Behring and the Kitasato showed that non-cellular fraction, which was the serum, has the protective effect. Ultimately, nowadays we know these are the antibodies. In 1880, Machenkov said these are the protective component is present in the cell fraction. And now we know that those cells are the T cells, B cells, neutrophils, phagocytes. Now we will go through what type of cells causes good immunity. But he proposed that concept, not just the non-cellular fraction, but also the cells imparts the immunity. Then 1940, Merrill Chase have shown, especially in case of tuberculosis, that transfer of white blood cells, and white blood cells are present in the blood. So you see the blood has, if you remove the red blood cells, remaining fraction of the cellular component is called white blood cells. Okay? And those white blood cells are the T cell, B cell, neutrophils, those kind of cells. Okay? And white blood cells are T cell, B cell, and K cells. And he proposed that concept that yes, if you take this cellular component which has the white blood cells, they can protect against the tuberculosis. And in your virus class, I mentioned earlier also that intracellular bacteria, for example, tuberculosis happens because of Mycobacterium tuberculosis. It's an intracellular bacteria. That means the bacteria has to go inside the cell, and the cell name is monocytes or macrophage. And inside the cell, and there it stays. And to kill this bacteria inside the cells, you need the white blood cells. Antibody do not work. Similarly, again, I will repeat that in COVID-19 time, this is again very important concept to know that despite if your vaccine is giving you lot of antibodies, okay, but it was not inducing the cell-mediated, cell-based immunity, those vaccines are not very successful. For virus, best vaccine would be the vaccine that give you lot of antibody plus cell-based immunity. And you may be listening now, the radio or the TV or reading online, that some of the time the current vaccines are not 100% effective. People are getting disease even after getting vaccinated. And the reason is exactly this, that those vaccines, they have been demonstrated that they develop a good antibodies. That's how they have been examined for their immunogenicity. But those companies, they have not performed an extensive work on cell-based immunity. They have done some work, but extensive work is missing. And maybe this is the reason that for the viruses and for intracellular bacteria, you need a vaccine that develops or that provides an excellent cell-based immune responses, then those would be more successful. I think this is a very, very good and important observation. All of you should know that there's only one viral disease so far against which vaccines were developed, and that disease is completely eliminated from the planet, or almost eliminated except at very few places. And that disease is the smallpox. Many of us are vaccinated against it, but none of us ever develop again because this vaccine is very successful. But this has got a very good history. Edward Jenner, who is very famous immunologist, okay, he observed in late 18th century that relatively mild disease of cowpox, which is a cousin of the smallpox. This mild disease cowpox has happened because of vaccinia. Okay, seem to confer protection against the fatal disease of smallpox. So if you vaccinate a person with the cowpox virus, which will only cause a mild disease, but if you vaccinate the person with cowpox, then that vaccinated person will never develop the disease against the smallpox. In 1796, Jenner demonstrated that inoculation with cowpox could protect against the smallpox, and he called the procedure vaccination. So the word vaccination came from the Edward Jenner in 18th century in year 1796. And from that time, people started vaccinating. And 1965 onwards, you see the number of countries with one or more cases per month. There were so many, and by 1976, 78, the disease was almost gone, and smallpox officially declared eradicated in around 1978 or 80. Okay, so after a period of 3 years in which no cases of smallpox were recorded, the WHO was able to announce in 1979 that smallpox had been eradicated, and vaccination was stopped afterwards in many, many countries, but in some countries still they do vaccination. Because of such an important subject in the medical biology, immunology got almost after 1900, once it is discovered, got popular, and it got almost more than 19 Nobel Prizes so far. Right from the blood group, ABO blood group was discovered by Landsteiner, then Emil von Behring, I mentioned about his work on the serum therapy or the plasma therapy, so-called the antibody basically discovery, or non-cellular fraction of the blood discovery, and then the matching curve for cell-based immunity. They all got the Nobel Prize. So what is immune system? It's a functional system, not an organ system, unlike the respiratory system, which is very defined that you have the nasal passage, then it goes to your trachea, to the lung. Similarly, gastrointestinal system, you can start from oral cavity, goes to esophagus, stomach, your intestine. Your cardiovascular system starts the heart. Unlike that, immune system is a functional system, not an organ system. It's a very complex system that includes, for example, our skin. Our skin is a part of the immune system because skin provides a physical barrier between the environment and your body. Nothing can permeate. Water cannot do it inside the skin. Germs cannot permeate from the skin. So it's a good physical barrier. Lining of the mucous membrane also provide you the physical barrier, right? Secretions like tears, mucus, they all have antimicrobial properties, so they can eliminate the first stage elimination of the microbes. Blood cells and vasculature, they have white blood cells, they are protective in nature. Bone marrow, which is inside the bone, this has again the regeneration capacity for the different white blood cells and red blood cells. Liver, our liver is very important because that makes complement proteins, and complement protein again they kill the microbes. Lymphatic system and lymphoid organs. So basically that's the system. You don't need to memorize all of it, but just want to give you a flavor that what is the immune system and how it is different from the organ system. Immune system, as I said, is a functional system and mainly made up of two different categories you can define. One is innate immune system, another is adaptive immune system. Innate immune system you can also think as non-specific. Non-specific in the sense that, for example, it will not differentiate between different types of pathogen. For example, it will not differentiate between bacteria, viruses, fungi, but we all have that first line of defense. And for example, skin doesn't differentiate between different classes of the pathogen, but act as a first line of defense. Okay, so that, for example, you can call it innate immune defense. Certain microbial proteins like tears you can call as first line of defense for the eyes. As long as something goes in eye, the first system will work and kill the pathogen. But second response is very specific, very precise, and that is adaptive immune responses, and that gives us a second line of defense. This is specific. If that is specific, for example, a response against the coronavirus would be different than the Mycobacterium tuberculosis like bacteria or the Candida, which is a fungus. So how a typical immune responses are generated. So you go from left, that any pathogen or invading microbes come to our body. For example, first it will come to skin or the mucosal membrane or to the secretion. They will act as the external defense. Correct? And then once they enter, let us say the first thing failed, then our internal defense mechanism, which is again part of the innate immune responses, and those are phagocytic cells. Phago means generally eating something. Okay, so the cells that can eat up, they are called phagocytic cells. So some cells in our body, they are called scavenger cells. They can just go simply eat the material, right? Or they can eat the microbes. Antimicrobial proteins. So I told you in the last class about the proteins. So we have the proteins that are having property of killing the viruses, bacteria, and those proteins are there in our body. They act as a first line defense and part of the innate immune responses. Similarly, other inflammatory proteins are also there. And the another type of cell, it is called natural killer cells, NK cells. NK cells, some immunologists think they are the part of innate, some things acquired, some things they are bridge. So you can, this can be up to you, wherever you want to put, but the name suggests they are innate or inherent properties killing something. So they are natural killer cells. Complement is another type of proteins. These are protein, but a cluster of group of proteins that work together to kill a pathogen. So this is first line of defense, innate immune response. Whenever innate immune system still couldn't kill the pathogen, a pathogen is still so strong that it passed through external, it passed through internal, then the acquired immune response or the adaptive immune response is generated. These are slower responses to specific microbes, and these can be divided into two parts. They again, acquired or adaptive immune response again can be divided in two categories. One is humoral response. Humoral means again the soluble component. Soluble means here antibodies. So either we will generate the antibody or we generate the cell-mediated response. That means those immune cells like T cells, cytotoxic T cells, especially for example corona, COVID is protect up nasal passage, for example, mucous membrane gets the virus. It passes through from mucous membrane. Now it has gone from, let us say, in your lungs. In the lung, all these cells are present, microbial proteins are present, inflammation response will also happen. But unfortunately, let us say all this fail. In majority of us, it is working. That's why we are all safe. But in some of us, it may not work. And then acquired immune responses are generated. So that's why I say slower. Slow means the reason it takes some time for antibodies to develop. And once antibodies are developed, they will fight. And similarly, the cytotoxic T cell mediated responses will also be generated. So this is slower, but it is robust. It is very specific. And this keep all of us, or majority of us, safe. First line of defense, innate immunity, you can say there is no time lag. As soon as the pathogen comes, we are ready to fight. It's not antigen specific, as I said again, and it has no memory. That means first time pathogen come, second time come, it will again act as it was working in the first time. It will not keep the memory that you came to me first time, I remember you, and then now you are coming, I will mount a big response against you. It doesn't keep any memory of the previous infection. However, adaptive immune response is very precise, is second line of defense. So there's a lag period because it takes some time to develop. It's very specific against the target, it's very specific against viruses, bacteria, or anything, but it also keeps a memory, development of memory. That means once infection happened, if the virus or bacteria comes second time, the previous infection memory is there, so that body now more prepared. There will be a shorter lag period. So this is very good concept for the vaccine again. COVID vaccine, for example. That's why they are giving everybody COVID vaccine, because vaccine will develop this adaptive immune response in you. And then whenever corona comes, then our body have already seen corona because of vaccine. So whenever real corona virus comes, the body will have the memory and immediately can fight against the virus and kill it. Innate immune system is external defenses. Basically, they're anatomical barriers. You can call them mechanical factors. For example, skin is act as a barrier. Mucociliary accelerators. So these are whenever you have, for example, cough. You will always see when you are having a throat infection, cough is always coming outside. If you see this mucociliary accelerator on the right, what you see, that mucus is sent outside. A flap is designed in a way in our trachea, in our tough pipe, you can say trachea if you can't remember, then it is sending upwards, indicating that this is not allowing infection to go inside the lung, but instead is throwing outside. That's why when you have the throat infection, you try to throw the cough out, to cough it out, right? So that's act as a good mechanism to protect our body. The flushing action of saliva, tears, urine. So when eye damage happens, a lot of tear comes, things are out. In the mouth, a lot of saliva comes during any infection, so that it protects. Urine also flush out any infection. Another important chemical factor that act as a innate defense mechanisms are present in our sweat. For example, when we sweat, antimicrobial peptides are there. So these are again the antimicrobial peptides which kill the pathogen. Similarly, our stomach pH is very low, almost like 1 to 2, and that kills majority of the microbes if we by any chance eat in the food. Lysosome in tears or saliva. So our tears and saliva again have the lysozymes, the enzyme that can kill all the pathogen or microbe that infect the eyes. There are biological factors that also protect us, right from your nose, from mouth, nose, skin, lungs, stomach, and intestine. We have so much normal flora in our body, more than 1,000 species of bacteria, and they compete with the pathogen for nutrient and space. And these bacteria, this normal flora keep us safe. So while I was teaching you bacteria class, I told you there are good bacteria and there are bad bacteria. There are given you even the example of E. coli that can make you sick, but E. coli are also good. So always remember that there are good bacteria which are normal flora, and good bacteria is extremely important. In fact, nowadays there's a huge, a very hot field of microbiome. A good microbiome is very important for a healthy life. So in our blood, for example, which is internal defense, we have the cells, different type of cells. And where are these cells present? These cells are present, for example, in the blood. So if you go to the IIT Bombay hospital, whenever you all come to the campus, you get and you go to the IIT hospital and get your blood examined, something like this report will come, which will tell you about HB means hemoglobin, RBCs, WBCs, neutrophils, then different type of lymphocytes, monocytes, eosinophils, basophils. What are these? It may come in your mind, what am I looking at? So these are your white blood cells, and within white blood cells, they are telling you what type of white blood cells you have, like neutrophils, lymphocytes, monocytes, eosinophils, basophils. So this is basically these cells act as the internal defense and they try to kill the pathogen. What are the source of these blood cells? Where are these cells are generated in the body? So blood cells are generally generated in the bone marrow, and from in the bone marrow there's cells called stem cells, which are hematopoietic stem cells. So stem cells can be very specific to certain cell types in the grown-ups. So adult stem cells, especially for the blood, these are the hematopoietic stem cells. And hematopoietic stem cells, they give rise to all different type of white blood cells, which you saw in the previous slide. So if you have hematopoietic stem cells, it divides into two parts, lymphoid cells and myeloid. And lymphoid cells give you the T cells, B cells, and NK cells, and dendritic cells. Okay? And the myeloid cells give rise to granulocytes, neutrophils, eosinophils, and basophils. So the major cell type of the adaptive immune system, or the immune system, which is very specific, those cell type are mainly the T cells and the B cells. B cells makes the antibody, and T cells take care of your all the specific immune responses. So, for example, if you want the COVID vaccine to work the best, you should ensure that B cells are getting activated and T cells are getting activated as well. B cells activated B cell will give rise to antibody. Activated T cell will give rise to the excellent T cell mediated protective immune responses. Okay, so in our body, the 20 to 40% of the body leukocytes are the B, T, and NK. They are basically present in the 99% in the lymph node. If they are inactivated, they are generally state in the resting state, and resting state of the cell is called naive cell, and they are very small, around 6 micron diameter. And this slide is just to let you know the sometimes nomenclature in biology becomes very, very difficult. And there are so many cell type T, B, NK, monocyte, dendritic cell. There's so many, sometimes one gets confused. So they brought the CD nomenclature. Personally, I feel CD nomenclature is also very difficult, but CD means a cluster of differentiation. And there are so many CD markers now, many based on the different cell types, so that they can be identified and classified properly. So even, for example, T cell, within T cell, they came up to many, many different subtype of the T cell. Now they call them CD4 positive T cells, or CD8 positive T cells, or CD3 positive T cells. So based on these CD markers, basically that's the proteins, which they call biomarkers as well. Based on these proteins, they identify the cell subtypes. Okay. The list is very long. I don't want you to memorize it. This is just for your information. There are so many markers, so CD1 to CD300, and they basically define the different cell type or the different state of the cell type. Now I give you the update on very brief update on different types of cells. Like I said, NK cells. NK cells are also called null cells. So these are the killer cells, for example, and they kill the virus infected cells. If in our body cells get infected with virus or cancer cell or a bacterially infected cells, these killer cells' job is to kill these cells. Okay, and you don't need to remember these very technical details, for example, but at least remember that natural killer cells, these cells are part of the innate immune system. They can be the bridge, and they can participate in the adaptive immunity as well. And they are the important cells as a first line defense for the differently, either for virus infected cells or cancer cells. Some of you may be thinking that how these natural killers know that this cell is a healthy cell and I should not kill it, but another cell is a infected cells or a transformed cells and I should kill it. That concept is there in the body. For example, I will not tell you in a technical word, but in a very layman language, natural killer cells, they will meet a healthy cells, and there are certain proteins which are expressed on this healthy cell. And when the NK cells see these healthy proteins expressed on these healthy cells, they will say, okay, you look like me, I am not going to kill you. But in the infected cells or in cancer cells, some cell surface proteins are missing here, or they are altered. And because of this altered surface expression of the protein, NK cells will realize that you are no more healthy cells and you should be removed from the system, and then it will kill. This is a mechanism how it differentiates between a healthy and infected cells or altered cells. Another is called mononuclear cells, and mononuclear cells could be monocytes in the blood. But when these mononuclear cells go to the tissue, they turn into the macrophages. That state of the cells is called macrophage. Same cell types, they both are monocytes, mononuclear cells. In the blood, the state is like called monocytes. In the tissue, they are called macrophages. And when these macrophages are present in different tissue, for example, in the lung, they are called alveolar macrophages. In the bone, maybe osteoclast. Similarly, in the liver, they are called Kupffer cells. In the nervous system, in the brain, they are called microglia. In the intestine, so different names are given based on their location. Okay? But these cells have a very good phagocytic activity. Whenever they see microbes, they will eat them up. Which you see on the top, both left as well as right, you see the phagosome. Phagosome is the vesicle where they have phagocytized the microbes. They secrete cytokines and antimicrobial enzymes, and using those cytokines and the hydrolytic enzyme, they will kill the microbes. Based on their tissue they reside, they are called different types. Activated by phagocytosis, or the different proteins in our body, they get activated and start killing the pathogens. So this micrograph tell you about how the macrophages phagocytose the bacteria in this case, and how they kill it, and what is the mechanism of that. So here you see this macrophages, which are pseudopodia. The bacteria comes, pseudopodia engulf it, and then it becomes phagosome. In the Professor Sanjeeva's classes, you have read about the lysosomes. Lysosome and phagosome fuse with each other, and that makes phagolysosome. And in the phagolysosome, these lysosomes, they carry the lysozymes, hydrolytic enzyme, and that hydrolytic enzyme will kill the pathogen. Another type of cells in the immune cells in the blood are present white blood cells. These are the ones you generally see in your hospital report, where you see the lymphocytes, neutrophils, monocytes, eosinophils, and basophils. And within lymphocytes, if you again further classify them, they are B cell, T cell, and NK cells. And these are the very important cells that fight against the pathogen in our body. And these are very important and critical cells of the immune system. For example, neutrophils, which I have shown you, they are the most abundant white blood cells. And in fact, 50 to 60% of the WBCs are the neutrophils, and they are the most important cells of the innate immune system. And whenever you have any damage or any inflammation, these are the cells which reach first to the inflammatory site. And what they do after reaching to the inflammatory sites, neutrophils, they do phagocytosis. Phago means eating something, cyte means these are cells that can engulf whatever they find, they eat and digest microbes. So these are the white blood cells. Their job is phagocytosis, and that's why these cells are called phagocytic cells. So how do these neutrophils eat and digest microbes? This figure shows that once the pathogen, which is in the purple thing, this comes in contact with the cells using pseudopodia that surround the pathogen, and then pathogens are engulfed by the process. The process is called endocytosis, because it's taking the pathogen inside the cells. And inside the cell, the vacuole forms. So it's not like easily floating. These vesicles, they're called vacuoles, they form. They enclose the pathogens, and these vacuoles and lysosomes, they fuse to each other. Once these vacuole and lysosome fuse, the toxic compound and lysosomal enzyme, they destroy the pathogen, and then debris from the pathogen is released by the process called exocytosis. So what is special present in these granules that kill the pathogen? These are the lysosomes, and they digest the bacterial cell wall and other microbial protein using those hydrolytic enzymes. Okay? These are the antimicrobial enzymes, and by the process hydrolysis, they kill the cell wall of the bacteria. Having given you a very brief about the immune system in a very superficial way, we are not talking in deep. Now, just want to give you a very brief idea about how the vaccines work and what is in the vaccines. So many times you know that vaccine, what they contain. Basically, vaccines contain the tiny fragments of the disease-causing organism. So, for example, either an attenuated form of the COVID virus, right? The killed virus, right? Or they can have certain blueprints for making the tiny fragment. For example, they can have the mRNA, which you learned from the gene expression class. Maybe they have mRNA, and then the mRNA, they make the viral protein, and that ultimately tell the body that virus has attacked us and makes the immune response. Or if not the inactivated virus or the blueprint like mRNA virus, they can be some other component of the virus itself. Proteinaceous, some proteins may be there, spike protein you will hear a lot in the newspaper. So some disease-causing, some fragments of the disease-causing organism, either organism itself or part of the organism, or the gene expression system like mRNA vaccines, those kind of blueprints. And they also contains other gradient to keep the vaccine safe and effective. So there would be this virus, attenuated virus, or the spike protein, or the mRNA of the virus, plus some solution or something, some solution that can protect that component, that can keep the that particular vaccine component in efficacious state. Okay. So these could be antigens, disease-causing organisms, and these are the key ingredient in the vaccine. And when these people are vaccinated with these vaccines, people feel that virus has attacked them, but it is not the real attack. They will not cause the disease, even if they cause disease very mild. But immune system get trained against those kind of fragments or attenuated virus or the mRNA virus. And here I have just summarized a little bit more about what are the ingredients. So antigens, that is active component which generates the immune response, and that antigen could be either the protein, it could be mRNA, it could be the virus. Then you have the preservatives that prevent the vaccine from becoming contaminated once the vial has opened. You have certain substance called surfactants. They keep all the ingredients in the vaccine and the blinded together. You have the diluent, which is a liquid used to dilute a vaccine to correct concentration. Adjuvant, that helps in vaccine important antigen to improve the immune responses. Then you have got stabilizers. So these except antigen, all other components are either to stabilize or preserve the protein for longer shelf life. And adjuvant meaning here is that they act as an improver of the immune responses in the vaccine. So for example, if you take two different type of vaccine, I would like to give this example because there's so much news about it. So you have the Covaxin, which is developed by Bharat Biotech here by the support of ICMR. And here the antigen is the inactivated virus itself. Okay, inactivated. And right now we know it is efficacious, and storage conditions based on its preservative diluent is 2 at 2 to 8 degrees, and we are giving 2 doses of the vaccine after first is 0 and after 2 weeks, and it's available right now in the country. However, the another vaccine is Covishield. All the research of Covishield happened by the AstraZeneca in the UK, and our Serum Institute is more like a contract manufacturer for this. So it is, I think, wrongly mentioned, developed by, instead you can say produced by or manufactured by Serum. And the vaccine type is the viral vector. So here they are using the non-replicating means the virus doesn't replicate, doesn't make more copies. But so that means the virus, actually the virus will not cause the infection, but this virus body will feel like it is cause that the virus has come and body will produce the immune responses. And this vaccine is also given 2 doses, and both the doses after the gap of 2 to 3 months. Okay, so with this, I will stop this basic immunology class. Hope you like it. And if you have questions,